And so now let me introduce Dr. Hayes. So Dr. Hayes received his bachelor's degree from Harvard University in 1989 and his doctorate from the Department of Integrative Biology at the University of California, Berkeley in 1993. Today, he is a professor of integrative biology at UC Berkeley, where his research focuses on the role of endocrine disrupting contaminants, particularly pesticides, and impact on global amphibian declines and public health. His research has resulted in a widely known study connecting the herbicide atrazine to the feminization of male frogs. From there, he has examined the exposure of racial and ethnic minorities to chemicals and the role such exposure plays in healthcare disparities. He has also been a continuous advocate for inclusion and diversity, particularly within higher education. And so with that being said, I'm going to go ahead and give him the floor.
Again, I want to thank you, Emily, for the invitation and for having me here. And when I think about, especially in these days, a lot of my invitations come from not only wanting to hear about the science, but also wanting to discuss diversity and my role as a Black scientist. And the first thing that many people might think of is there is no such thing as BIPOC science or Black science. Science is science. And so how are the two connected? And I think there are many ways, especially if you're an underrepresented person of color, that even though science is science and we all do the science the same way, the idea and the concept of who you are and race pervades in many interesting ways throughout the science. And so what I put together today, I'll watch the time, and I don't necessarily have to get through everything. We can just, you said 30 minutes and we can just shut off at 30 minutes, especially since we've lost time. But what I thought I'd do is give my same old research seminar, but actually make a point of places where I think that my background and being a person of color has affected the science and affected my career, and that'll open us up for a discussion that I think we want to have. So, I'm going to tell you a story, and before I get into the meaning behind the title, From Silent Spring to Silent Night, A Tale of Toads and Men, I think we can start right here with a thing that sort of who I am and where I come from has a big impact on even how I present my science. I always start my talks with the acknowledgements first. I am very keenly aware that it took a lot for me to get here, and I'm thankful for the people who made it possible for me to be here and who made me who I am. And I always thank those people before I present my science. First and foremost is my family, and this is an old picture of my immediate family back in South Carolina, but I owe a great deal to my mother and my father, Romeo and Susie Hayes, for their love and support over the last 53 years. My wife, Catherine Kim, and my son and my daughter for their love and support. I also have to acknowledge the funders that have made my research possible. As you just heard, I've spent a large part of my career working on atrazine, and I was originally funded to do that work by the chemical manufacturer Novartis, Syngenta. So this is a matter of disclosure for me as well. And I also want to thank the students. This is a current photograph of, fairly recent photograph of my laboratory. I've had an incredible experience working with a diversity of students of every kind of diversity that you can imagine. And they've given me very different perspectives on science and the importance of science and the importance of communicating science. In particular, because I study agrochemicals, I've been just had a wonderful experience working with people whose family worked in the agricultural industry. And so studying chemicals and their impacts on animal health and wildlife health and public health, and working with students who then would say, hey, my mom and dad have been sprayed with this chemical, gives me a completely different perspective. And finally, I want to acknowledge my grandmother, who was like a third parent to me, who passed on, luckily for me, passed on her love of education and her desire to make the world a better place through education before she herself passed on. And she's also the person who taught me that if you want to get a point across, don't preach, don't teach, just tell a good story. And that's why I opened up today telling you that I'm just going to tell you a story. And my story, in fact, starts at my grandmother's house, shown here. My grandmother was born in that house. Well, first and foremost, my grandmother's grandfather built that house. when my grandmother died, the bill of sale for that house and for my grandmother's grandmother were still in that house. My grandmother was born in that house. My mother was born in that house. And so a lot of my history and a lot of who I am were in that house and go back to my childhood in South Carolina. The other thing that's important for the story is that it's also at my grandmother's house where I fell in love with biology. My parents used to drop us off at the house on weekends to spend the weekends with my grandmother. And she had a huge, or what I remember to be a huge forest behind her house. And we spent hours lost in my forest. So maybe she wasn't the best babysitter ever, but we spent hours lost in that forest behind her house. And here I can show you what that looked like. There's the house, and that was it. That was the whole thing. In my mind, though, in my memory, it was as big as the Amazon, and we spent hours there, and it was there that I watched birds build nests, that I watched eggs being incubated and hatched in bird nests, that I watched snakes and lizards vying for territories and fighting with other conspecifics. And it's where I watched frogs lay their eggs and where I fell in love with amphibian biology. And that's, I always, in my talks, present myself as just a little boy who likes frogs. That's where my love of biology came from. My mom sent this book to me when my son was born, and she wrote a note in there that said, "This was your dad's favorite book," a note to my son. And I don't remember the book, but that history and that experience in my childhood has been with me all of my life. And still, with degrees behind my name and everything I've been through, I'm still ultimately just a little boy who likes frogs. One of the other important events in my professional career was getting to go to Africa and to do field work in Africa. And again, that also reflects on my childhood. I remember my father bringing home National Geographic magazines, and I remember folding out the maps and dreaming of going to this magical place. And I got that opportunity to go to Africa and to do research when I was in graduate school. And not only did I get to go to Africa, but I was funded by National Geographic. I got to be in the show. I got to be in the magazine. I got to, to literally live out my childhood dream. If you can imagine, I remember as a kid watching National Geographic television and watching that guy and going, gee, I want to be that guy. And I got to be that guy. So if you can imagine your childhood dream coming true in the truest sense of the word, that is what I experienced by the time I got into graduate school. While I was working in Africa in a place called Arabuku-Sokoke, which is here on the coast in Kenya, I discovered a species. Well, I didn't discover the species for the first time, but I discovered that a species, Hyperoles argus, that was sexually dimorphic. The males look like they don't belong to the females. In the laboratory, we discovered, and we did discover for the first time, that they all start out green, but the females change color at sexual maturity. We hypothesized and showed that the hypothesis was correct, that color change was controlled by estrogen. The same by estradiol estrogen, the same estrogen that's in every vertebrate animal, in every reproductive female of every vertebrate animal known. And from there, we ended up in an interesting situation. We ended up patenting that frog, and we patented it for the following reason. So here's a control. They all start out green at metamorphosis, as I said. If we dip them in estradiol, we discovered that we could force the color change. We can induce premature color change, and we can induce even genetic males with testes to start to develop a female color pattern. We also showed that we could dip them in ethinyl estradiol. This is a synthetic estrogen that's used in birth control pill, and they would change color. We showed that we can dip them in DES, a pharmaceutical estrogen that's used in humans, and they'll change color. And we showed that we can even dip them in pesticides like DDT, which is not an estrogen, but it's an estrogen mimic in humans, and it also made them change color. In fact, we tested dozens of compounds. Sorry? No, I think that was a mistake. So we screened dozens of compounds, and we showed that every estrogen that was known that made my frog change color was also known to promote breast cancer. What's more is we showed that we could treat with the estrogen blocker tamoxifen and stop the color change. So not only were we able to identify compounds that are estrogenic in my frog that might be associated with promoting breast cancer in humans, but we also showed that we could do a pre-screen or test chemicals that block estrogen in my frog that might then go on to be used to treat breast cancer in humans as well. So with that, the university, of course, it's their intellectual property, and the university said, well, how are you going to make money from this? Either you show us how you're going to make money for it, or we're going to sell the intellectual property. And it was there that I got involved with what was then Novartis, and they gave us a contract and asked us, well, can you test atrazine and determine if atrazine is not a hormone? At the time, it was not known as a hormone mimic. It's an herbicide or a weed killer that's mostly used on monocot crops. In this country, it's mostly used on corn. It's been used since 1958, and we use 80 million pounds per year in the United States. At the time, it was the #1 selling agrochemical in the world. It's used in more than 80 countries, so it's fairly widespread, and it's now outlawed in all of Europe, and with the irony being that the company is based in Europe. So here we are in this country using 80 million pounds of a chemical that's not allowed on the continent where the company calls home. It's produced here, but the company is based out of Europe. So we worked with African clawed frogs, although we collect them in San Diego, they're invasive. So I call mine African American clawed frog. But we worked with African clawed frogs, the sort of stable model amphibian used in biology. We showed that atrazine inhibited growth of the voice box in males. So, and this was an indication that atrazine was inhibiting testosterone, because male frogs sing and females don't, for the same reason that in general men have deeper voices than women. So again, there's a endocrine connection between my frogs and humans. And if there's a problem with testosterone, of course, you go to the source, and the source of testosterone is the gonads. And we showed that a portion of the animals exposed to atrazine had gonads that didn't develop typically the way that they do in amphibians. This is an animal, if you're not used to looking at frog gonads, that has testes, then it has ovaries, then it has more testes, then it has more ovaries, which is not typically how amphibians develop. There are fish that are naturally hermaphroditic, but this is not typical of amphibian development. And so we propose that the following would explain our observation. Normally the testes, represented by this big yellow oval, makes testosterone. In fact, the word testosterone literally means testicular hormone. It's a portmanteau. And we propose that atrazine turned on the enzyme that converts testosterone into estrogen. Estrogen literally means the generator of estrus. It's the so-called female hormone. And as a result, the animals are demasculinized, like their larynx doesn't grow, and they're subsequently feminized because now they're producing estrogen. And if you're a genetic male, you shouldn't be producing substantial amounts of estrogen. We went on to show that this hypothesis was supported, that here are control adult males, and these are actually daytime testosterone levels. If you give them atrazine, testosterone is reduced, and to a point that it's not significantly different from control females. We published that paper in Proceedings of the National Academy of Sciences, hermaphroditic demasculinized frogs after exposure to the herbicide atrazine at low ecologically relevant doses. And this was significant for a few reasons. It's significant to me personally. One is 4 Black men and a Latina co-authored the paper. It's probably a record for PNAS. The other one is it was a significant publication in my career because I was approaching tenure, and getting a well-reported paper in a journal like PNAS was pretty helpful into me getting tenure. It's also significant in another way. I remember calling my mom when this paper first came out and saying, Mom, I have this really important paper coming out in this critical time in my career. I have a paper coming out in PNAS. And I remember my mom, first of all, going, wait, you have a paper cut on your penis? I'm like, no, PNAS, Mom. And then I remember her calling me back the next weekend and asking, how important is that paper? I went to Barnes & Noble. They never heard of that magazine. And it's another reflection on me and who I am and where I came from, in the sense that the things that we do in the ivory tower mean nothing to 99.999% of the world. My most important publication is in a magazine that my mom can't get access to. And so it started to make me rethink about what my role is in, um, not just as an academic and a scientist, but as a citizen and in the world in general. At any rate, as important as that paper was, it didn't answer a couple of questions. So it was significant scientifically, but we didn't know if these hermaphrodites that we discovered were males with ovaries or females with testes. And we also didn't know what happens when they become adults, because we only looked at the exposed larvae after metamorphosis. We didn't grow them up, which takes 4 or 5 years for the species. And I'm just going to tell you very briefly, because I want to end early enough so that we have lots of discussion. But in brief, about 8 years after, because it took us that long to actually figure out all the questions that we wanted to answer, we discovered, one, there was a gene discovered that is associated only with females, a gene called DMW. So that's what's shown here. This is just standard PCR. And what we were able to show is that of the frogs exposed to atrazine, about 10%, once they were adults, completely transformed into females with ovaries, even though they were genetically male. So this shows a genetic male who looks like he's smiling, and he's actually doing exactly what you think he's doing there. And this is actually a sibling that's also, as you can see, a genetic male. But this particular animal had ovaries and laid eggs, just as if it were a genetic female. So about 10% of the genetic males that were exposed to atrazine, that were presumably hermaphrodites in metamorphosis, completely grew ovaries and became females as an adult. And then I'm skipping a lot of the data, but we did studies to try to ask, what about the atrazine exposed male that didn't grow ovaries and become females? It turns out that they are unsuccessful. Atrazine exposed males are unsuccessful at competing for females in a group setting, but also, even if we pair them with females, they have very low fertility. So this shows that assay. This is about 2,000 eggs that a female releases. And what we did was we collected, after population, we collected the eggs and we counted the number that were unfertilized, shown here in the top left panel, side panel. And we counted the number that were fertilized. So these are going through gastrulation and neurulation and elongation. And essentially we were able to show that control males fertilize about 85% of a female's eggs, whereas atrazine exposed males only fertilize about 15% of a female's eggs. And that's in part because they don't show male typical breeding behavior, so they don't try. But it's also in part because the testis, in the absence of testosterone, does not produce adequate sperm. So this is a histological section through the gonads of a control. And you can see here that the testicular tubules are filled with sperm. So these are the so-called sertulae or nerve cells around the edge. The dark staining purple are the sperm. And the atrazine-treated animals, most of the testicular tubules have cellular debris, and most are absent of any mature sperm. So we published a second paper called Atrazine Induces Complete Feminization and Chemical Castration in Male African Clawed Frogs. The company hates the term chemical castration. That's why I put it in the title. And this was also published in PNAS. But even more significant, 9 undergraduates co-authored this work, and every one of them now has a PhD or an MD, and in some cases both. And I'm equally proud of that as I am in science. So now moving away, or getting away from the laboratory a little bit, we did studies where, and actually I'm still analyzing the data, where we collected frogs all over the United States. And we were able now to compare the impact of agriculture and atrazine on these sex development problems that are induced by atrazine on animals in the wild, to try and address the impact of atrazine in the real world. To bring this back home, Kangaroo National Park is the only national park in South Carolina. And in fact, when I grew up, it wasn't a national park. When I grew up there, it was just a swamp that I hung out in and collected frogs at. So it's one of the places where I learned to fall and where I fell in love with biology. A few years back, an article came out, an exclusive, showing that chemicals in medicines tainting Kangaroo National Park waters. And it turns out that the chemicals that they're talking about are, you guessed it, atrazine from agriculture in South Carolina, and ethynyl estradiol from leaky septic tanks in the neighborhoods around the park. This entire article here focused on, what about the weekend warrior who comes in to, kayak through the park? What if they fell over into the water? When in fact, of course, I immediately thought about, well, what about the frogs that are exposed to ethynyl estradiol and atrazine, two chemicals that I study? What about the impact on the animals that the park is put there to protect in the first place? And two, forget the weekend warrior. What about not forget the weekend warrior, but I'm more concerned about the low-income Black community that lives around the park and they're exposed to those chemicals year-round. The reason that the ethynyl estradiol is there is because of the leaky septic tanks in those low-income communities. So this is another case of where I've always joked that I took the hood to Harvard. This is a case where I get to bring Harvard back to the hood. So now we have a project where we're studying the impact on environmental health and public health of the chemicals that are now tainting Kangaroo National Park. Another thing that I think you find with people of color is often the desire to give back and to give something back. And I think that's manifested in my interest and my excitement about doing that study. I'm also more concerned about environmental pollutants and their role in the declining amphibian population. So for those of you who don't know, 80% of all amphibians, the animals that I fell in love with as a little boy who likes frogs, are in decline. And I'm not going to tell you that it's because of atrazine or chemicals in general, but this is from a paper I published with graduate students a few years back, showing that the main cause of amphibian decline is habitat loss. But if the habitat is lost and the only places amphibian you have to breed are agricultural areas that are contaminated with pesticides that cause abnormal sex development and that impair immune function, then those pesticides, those chemicals play a critical role in those amphibian declines and likely a critical role in impacting environmental health for humans and impacting public health. Now, this is a slide of a former student of mine that started working with me back when I first started. She is now, Nadine Burke-Harris, she is now California's first ever state surgeon general. So we've made history a little bit there. And this is a book that she published on her life and how she got to where she is. And she cites in that book, I'm not, of course, not taking credit for her work and her success, but she cites in the book that she became well known for studying the impact of crowding and stress and chemical exposure on low-income Black communities in California. And she cites there, you can see she's citing my name, Tyrone Hayes, but she got the ideas for her work and the impact of crowding and stress on health, that she got those ideas for studying children that way from her work in my lab, studying the impact of crowding and stress and chemicals on tadpoles. So just one connection. I like to show this photograph to illustrate the oneness of environmental health and public health. And it's a figure I took in Lake Nabugabu in Uganda, where the runoff from this crop, which goes into these containers, is the sole source of cooking and drinking water for this village. And I used this slide to point out that if we identify problems associated with this runoff from their food, in this case, if we show problems, developmental and immune problems with animals living in that runoff, then you might expect there might be similar problems in the human population that's actually also consuming that tainted water. This is a photograph of Oakland, where I live. I live somewhere here, and I don't have to collect my water in yellow containers. And even though we consider that because of EPA and other agencies that there would be nothing bad in our water that would negatively impact us, all you have to do is ask somebody in Flint, Michigan to know that that's not always the case. So I've become very concerned about how the role of chemicals in amphibian declines in what I call the silent night might be impacting human and human health. So in the same way that Rachel Carson in Silent Spring taught that the death of birds and the role of pesticides was a warning to humans, I very much feel that the new canary in the coal mine, our frogs, and our pending silent night is also a warning to humans that we should be concerned. Now, this is a quote from a colleague of mine who wrote, in ecoepidemiology, the occurrence of an association in more than one species and species population is very strong evidence for causation. I've told you about not just correlative experiments, but controlled experiments across population, species, genus, and I actually haven't told you, but we've also worked with other species in other families of frogs. But I've also now collaborated with scientists from around the world who've shown that these effects occur in all vertebrate classes. I published a paper with 22 scientists from 12 different countries, and we didn't know each other before this at all. And we discovered, though, that in our studies of atrazine, we've shown very similar effects. And I have to admit, in part, I drummed up this collaboration because the company has started to attack me professionally, claiming that my science was poor and not repeatable, and that I was the only, that it was just this crazy guy from Berkeley on frogs. There was nothing else to worry about. Well, what we were able to show is that here's my frog testis with sperm. As I showed you, give him atrazine and there's no sperm. Another scientist in England showed the same thing in Belgium. So here's fish, sperm in the testis, give it atrazine, no sperm. Here's a work from Argentina on reptiles. This is a caiman, it's like a big alligator. Sperm in the testis, give it atrazine, no sperm. Next is work that was done in both Croatia and Nigeria in rats. Sperm in the testicular tubule, give it atrazine, no sperm. And this work was done in birds and quail in Pakistan. So the effect has been shown in every vertebrate class in labs, independently in labs around the world. What's more is, do we see a decline in sperm because testosterone is declined? Here's work in fish. This was done in England, in salmon. Here's my work in frogs. Here's work in rats that was actually done by company-funded lab. So the decline in testosterone was repeatable in animals around the world. Now when we come to humans, we can't do experiments, but my colleague Shada Swan showed with a significant p-value that men who have measurable atrazine in their urine have trouble getting their wives pregnant and a low sperm count. They have about 0.1 parts per billion atrazine in their urine is associated with the low sperm count. And for your reference, 0.1 parts per billion, 0.1 micrograms per liter is the level of atrazine that we use in my laboratory to chemically castrate frogs and fish. What's more is, and now I've squashed the data down, but I've left the y-axis there, it's because here are atrazine levels in men who work in the field, and now I'll squash it down again. And here are atrazine levels of men who apply atrazine in the field. Men who apply atrazine have 24,000 times the atrazine in their urine than we know is associated with adverse effects on reproduction and fertility in those men. Men who apply atrazine have 24,000 times the atrazine in their urine than we use to chemically castrate frogs and fish. To put this in perspective, I could collect urine from men who work in the fields applying atrazine, and I could dilute the atrazine in their urine 24,000 times, and I could use the atrazine in their urine to chemically castrate and feminize 24,000 tanks of 30 tadpoles each. What's more, as we explored the other half of the equation, does atrazine induce aromatase and produce estrogen in other animals? Now in fish and amphibians and reptiles, it's going to cause phylogenesis and oogenesis, yolking up and egg production. But in mammals, we'd be concerned about breast cancer and prostate cancer. In that regard, with regards to prostate cancer and with regards to breast cancer, there are significant findings. There's a study in women whose well water is contaminated with atrazine with a p-value 0.0001. For those women who use that well water as drinking water, they're more likely to develop breast cancer. There's a mechanism for that. We've shown, and at least one other study has shown, that if you take human cell lines and expose them to atrazine, they'll start expressing aromatase and making estrogen, just like we've shown in amphibians, just like we've shown in fish, just like we've shown in frogs and reptiles. That's significant because breast cancer, such as this damaged cell I represent here, depends on estrogen, and the local production of estrogen via aromatase is important in stimulating those damaged cells to turn into tumors and to spread. So much so that the #1 treatment for breast cancer is a chemical now called letrozole. And letrozole works by reducing aromatase and reducing estrogen and preventing those damaged cancer cells from turning into tumors and from metastasizing or spreading throughout the body. The interesting thing is that atrazine, of course, does the opposite. It turns on aromatase, it increases estrogen, and it promotes breast cancer in rats. And as I said, it's associated with breast cancer in humans. What's interesting is Novartis, the company that originally funded me and the company that I worked with for the first 3 years of these studies, offers treatments for cancers that range from breast cancer, et cetera. So the same company that produced atrazine produced letrozole. So if you were living in the Midwest taking letrozole to fight your breast cancer, it was fighting it out with the chemical made by the same company. In fact, the #1 product of that company at the time, which did exactly the opposite. I've been to visit them. They're now called Syngenta. And just to give you an idea what the place is like where they produce this stuff, this is in San Gabriel, Louisiana. There's a pipe that flows directly into the Mississippi River, and 1.2 million pounds of atrazine flows in the Gulf of Mexico every year. It's a low-income community. It's 80% Black or African American, most of which looks like this. And I bring this up because, again, I'm just a little boy who likes frogs, but now I have this concern for environmental justice and environmental racism. As you may know, these are the top 13 cancers that you're more likely to get if you live in the US. And in red now, 11 out of the 13 are the ones that you're more likely to get if you're Black, if you're African American. You're also more likely to die from 13 out of 13. And this is a situation that cancer is an environmental disease. My colleagues who are experts on cancer say that less than 30% of cancer can be explained by genetics. So when your doctor tells you that you're more likely to get and die from cancer if somebody in your family has it, they're not telling you you have bad genes. They're telling you that you've been exposed to the same crap as the rest of your family. And as a minority or an immigrant or someone from low-income community, you're more likely to live and work in areas where you're exposed to chemicals that we know are associated with adverse health outcomes. So I think what's happened is that my interest in this aquatic organism has taught me a great deal about this aquatic organism, even though I didn't set out to study humans. We now know that your children will be exposed to over 300 synthetic chemicals before they leave the womb, with the high end of that exposure being if you're low income, if you're a minority, if you're a person of color, if you're an immigrant. And most of those chemicals, we have no idea what they do. I would argue, though, that a fetus trapped in a contaminated amniotic fluid is not all that different from one of my frogs trapped in a contaminated aquaria or a contaminated pond. And then lastly, in the last few minutes, and then I'll stop, is I want to show you a few studies that have moved me more than my studies on amphibians. These are studies that were done that are peer-reviewed and published in the literature on rats. And by the way, I've told you that I study frogs because I'm a little boy who likes frogs. I'm pretty sure that people who study rats aren't little boys and girls who like rats. They study rats because they tell us something about humans. Atrazine causes prostate and mammary cancer in rats. And again, rats being a proxy for us as mammals. Atrazine causes immune failure in rats, which we've also shown in frogs. Atrazine causes neural damage when rats are exposed in utero. Atrazine, and this series of studies were done by the Environmental Protection Agency, peer-reviewed and published, and they're the ones who impacted me the most. Rats that are exposed to atrazine are more likely to have an abortion. Of those rats that don't abort, the sons are born with prostate disease. Right? They're born with the prostate of an old man. Of those rats that don't abort, the daughters are born with impaired mammary development showed here, such that when they grow up, their offspring have retarded growth and development. And what strikes me about this study is that this rat on the bottom never saw atrazine. The rat on the bottom was affected by atrazine that its grandmother was exposed to. And we're seeing effects in our frogs now 7 generations down from the exposure. And again, these sort of epigenetic and transgenerational studies, there's several with atrazine in rats. And it makes me realize, and here's my daughter when she was young, the idea that my grandkids, that your grandkids could be affected by chemicals that we're using today moved me much more. It made me realize that I have much bigger responsibility than just a little boy who likes frogs. This is a figure showing that if you get pregnant doing peak atrazine contamination, you're more likely to have children with birth defects. Those birth defects include, among other things, genital malformations in male babies. And so this isn't my study, this is a human study. And I apologize, there are graphic images coming, but the study showed that if you're exposed to atrazine and you're pregnant with a son, you're more likely to have a son with hypospadias, where the urethra doesn't end all the way through the penis. You're more likely to have a son with cryptorchidism, where the testicles don't descend into the scrotum. And you're more likely to have a son with micropenis, where the penis doesn't grow. And I picked this study to show because male genital development depends on testosterone, and all of these malformations can be induced by excess exposure to estrogen. And if you're exposed to atrazine, a chemical that reduces testosterone and increases estrogen, and every vertebrate animal that's been examined, you have a baby that looked like it didn't have enough testosterone or that was exposed to too much estrogen. I show this figure because also most of the toxicity and safety limits for pharmaceuticals and agrochemicals are based on exposure to an adult white male. This is actually a guy who doesn't like me. He works actually for the company. That's why I use him in the figure. The EPA has recently acknowledged that atrazine is a reproductive toxin, but they reported to New Yorker magazine that a monetary value is assigned to disease impairments and shortened lives and weighed against the benefits of keeping a chemical in use. My concern is that atrazine is not being used to grow more food. As you can see, this is a graph of where all the corn grows in the US. And what you might notice is missing from the map is food. This feed here is what they use to feed animals, not what goes to humans. We eat less than 2% of the corn we grow, and atrazine increases corn yield by only 1.2%. So that price that people are paying that the EPA mentioned is not because we're producing food for people. What's more is we know that the price is paid not by the same people that are reaping the economic benefit. And I'll show this one last figure to illustrate that. 95% of our food is grown by Latinx individuals. These aren't the people that are reaping the money. If you look at Australia, if you look at California, we're the 5th largest economy in the world because of agriculture. In fact, 50% of the US's food comes from California. Half of the US's food comes from California. As a result, we use more pesticides than any other state, and 90% of the workers are Latinx. If I show you in red now, the top 10 counties for agriculture, the 30% of the land that makes us the 5th largest economy in the world, and if I plot on top of that the 30 poorest towns in California, there's an overlap. So the people who make us the 5th largest economy in the world are the people who are paying that price for the people who make money on selling that food. And again, I think as a person of color who works with people of color who work in that industry, even though my science is the same, and I'll stop, I have a few other slides, but I'll stop to share now because we're at 30 minutes. The people who make the money aren't the same people that are being impacted. And I've become very interested in the environmental justice and environmental advocacy as part of my research. In part, I think because of my background, but also because of the background of the students that I predominantly work with. And I'll stop there because I know we want to have a healthy discussion. And if other things come up related to the slides, then we can address those. But thank you for your time and attention.